All right, why don't we get started? So thank you, everybody, for coming to the CTSI seminar series this week. There are 5 more till the end of the year. I think in particular, we have Peter Crompton, so Joe Crompton's brother from the NIH, who will be speaking here in a couple of weeks. He actually has this really neat story on anti-malarial vaccine. But today we have the pleasure of having Dr. Yvonne Chen speak today. It's a pleasure, obviously, for me as well, because I count her as both a colleague and a friend. But as many of you know, she is an associate professor of microbiology, immunology, and molecular genetics here at UCLA. She did her undergrad education at Stanford, majoring in chemical engineering. You'll see this as a theme. She went to PhD at Caltech, did a PhD in chemical engineering. After she finished her PhD, she moved up to Seattle to do a postdoc fellowship at Seattle Children's with Mike Jensen, designing new bispecific chimeric antigen receptor constructs. She then moved out to Harvard Medical School for further postdoctoral training in the Department of Systems Biology. And then she came back west. She started in 2013 her own lab here in the Department of Engineering, but later moved over to microbiology, immunology, molecular genetics. She's now the co-director of the Johnson Comprehensive Cancer Center, together with Tony Rebus, and a member researcher in the Parker Institute for Cancer Immunotherapy. I think as you'll see today, since she has started on faculty, she has really developed into a leader into this CAR T-cell space. I think you'll get to see all sorts of these aspects in this research world. She's been invited to speak all over the world on this subjects, and is even involved in the creation of a new company to market her CAR T-cell designs. She's published widely in this field and has a multitude of high-impact publications spanning from both the preclinical, the basic side, both to the clinical side. She's designed new bispecific CAR T-cell constructs targeting CD19 and 20. And as you'll see, these are likely to be considered the best class CAR T-cells for B-cell lymphoma in the near future. But she also has new CAR T-cell designs for multiple myeloma, and we're even trying to bring her into the brain tumor sphere to get CAR Ts for malignant brain tumors. She's designed chimeric antigen receptors that for rewired responses to soluble ligands like TGF-beta. And I think for the trainees here, I think she's a great example of a basic scientist that sits right at the interface between basic and clinical science and medicine. And it shows you how relevant these ideas are for adoptive T-cell transfer in patients and in preclinical models, and how they can go between. So today she's going to talk about some of this work, and the title is Engineering Next Generation CAR T-cells for Cancer Immunotherapy. Please give her a warm welcome.
Thank you, Rob, for that kind introduction and for promoting me to the co-director of the Cancer Center. I'm just the co-director of the Tumor Immunology program within the Cancer Center. And thank you all for being here. I'm actually kind of surprised how many people showed up in person, because usually when it's hybrid, I talk to an empty hall. So thank you all for being here, and very happy to have the honor to present to you today. As Rob mentioned, my lab works on engineering CAR T-cell therapy for cancer. And just so I do not get screamed at by our conflict of interest committee, I will show you my disclosure slides. Some of the technologies that I'll discuss with you today have been licensed to Impact Bio. So I think many in the audience are already very familiar with the idea of adoptive T-cell therapy, where instead of giving patients chemo, radiation, or surgery, we give them living white blood cells, T-cells in particular, as the cancer drug. And what we usually do, this is in the context of autologous T-cell therapy, we isolate T-cells from the cancer patient, we genetically modify them to express a tumor targeting receptor. We primarily work on chimeric antigen receptors, or CARs. We then expand these engineered T cells ex vivo prior to reinfusion back into the patient. And this has worked quite well for a variety of B-cell malignancies, but there's still quite a few challenges that still need to be overcome. And so my lab is particularly interested in understanding, can we use biomolecular engineering and synthetic biology to make better T cells? And I generally describe them as smarter and stronger, smarter in the sense that we need these T cells to accurately identify tumor cells and distinguish them from healthy tissue that we do not want to harm. And stronger in the sense that once the T cells find those tumor cells, we need them to be able to efficiently eliminate those tumor cells. And that involves not just strong sort of effector function, but also the ability to overcome a variety of evasion mechanisms that tumor cells have come up with. And so, as I mentioned, we're primarily interested in CAR T cells, and CARs are these sort of Frankenstein fusion proteins that connect ligand binding domains derived typically from antibodies, although they don't have to be, connected to extracellular spacers, and then transmembrane domains. And finally, CARs typically have CD3 zeta, which is a dominant signaling domain in naturally occurring T cell receptor complexes, coupled in second and third generation CARs to co-stimulatory domains, most commonly CD28 and 41BB. Now, these CAR molecules are what I think of as single input, single output signal processing units, by which I mean they look for a single antigen, for example, CD20, that's present on target cells. And when they find those antigens, they try to kill the target cell that presents that antigen. Okay, now I'm grossly oversimplifying what's actually happening here, but this basic concept has a number of very important advantages. One is that because the T cells are engineered to target specific antigens, as opposed to say all the cells that are dividing rapidly, they're much more specific to tumor cells. And because these are living white blood cells, they can achieve systemic mobility, where they can circulate throughout the body and potentially identify tumor cells, even when we didn't know where every single lesion was located, right? And if you program these CAR T cells correctly, they can serve as serial killers. So Dr. Carl June's group had done an early clinical trial at UPenn with CD19 CAR T cells and estimated that each one of the CD19 CAR T cells could kill upwards of 1,000 leukemic cells inside the patient. And finally, and this is a property that's very important to engineers, these CAR molecules appear to be quite modular. So the different functional domains I described for you earlier can often be replaced by alternative parts. So for example, I can take a CAR that targets CD20, I can chop off its ligand binding scFv domain and replace it with a different scFv, one that say binds HER2. And then I'll have a HER2 CAR. And that new CAR is very likely to have activity. Okay, but I think it's important to note that just because something has activity doesn't mean it's good enough to have therapeutic function. And so a lot of times what we end up doing is a lot of molecular tweaking to get the molecule to be sufficiently robust to have therapeutic value, right? Now, despite these advantages, there's still a number of challenges, as I mentioned before. Today, hopefully I'll have time to talk to you about the first two, antigen escape and immunosuppression. There are additional challenges, for example, particularly for solid tumors. It can be hard for the T cells to even find the tumor, right? And so how do they traffic to the tumor? Furthermore, oftentimes tumor cells don't have anything that's uniquely a tumor antigen. So oftentimes we're targeting tumor-associated antigens that are also present on healthy tissue. Then we can end up with on-target off-tumor toxicity. And finally, there are certain adverse events that have been associated with CAR T-cell therapy, such as cytokine release syndrome, neurotoxicity, et cetera. And we'll touch on this briefly, but there are various strategies that we and others are exploring to try to overcome these. But I'll start with antigen escape, which refers to the situation where the tumor escapes from therapy by losing the antigen that the T cells have been engineered to target. So CD19 CAR T cell therapies were the first to get FDA approval. It's now approved for a variety of B cell malignancies, including lymphoma. So here I'm showing you a registration trial data for two different products, Yescarta and Kymriah, for the treatment of lymphoma patients. And what you can see is this long tail of approximately 40% long-term tumor-free survival, which is very remarkable if you consider the fact that these are patients with at least 2 or more lines of prior therapy. So they didn't have a whole lot of good options by the time they enrolled in this trial. But what we also see is that the majority of these patients eventually relapse, and they tend to do so within 6 months. And because of that, the median progression-free survival was only 3 to 6 months in these registration trials. What was also reported was that a substantial fraction, not all, but a substantial fraction of patients who do relapse end up with tumors that no longer express CD19. And that's what we mean by antigen escape. Once the tumor loses CD19, they can evade detection, and therefore the CD19 CAR T cells can't do anything about them. And so we started thinking, how does one overcome this problem? And a fairly, again, simplistic idea is, well, why don't you engineer T cells to target more than just CD19? Okay, CD20 is also a great target. In fact, it has a much longer history as a clinical target for B-cell malignancies. It's like CD19, a pan B-cell marker present on all mature B cells prior to differentiation into plasma cells. And, we have decades of clinical data with anti-CD20 antibodies like rituximab that tell us even after patients become resistant to antibody therapy, they rarely become completely CD20 negative. It can happen, but it's quite rare. And so that suggests to us CD20 can be a good partner to CD19 because it appears to be more resistant to antigen escape. So how do you make a T cell recognize both? Well, we decided to build what is now described as a tandem CAR, basically the canonical CAR architecture with an additional ligand binding domain connected in tandem. This work was primarily done by a former PhD student, Eugenia Zha, with help from another former student, Mengyin Lin, particularly on the in vivo data I will show you. Now, this work has been published for quite some time, so I won't go into the details of how we engineered the CAR, except to say that the specific amino acid sequence, the rigidity, the size of the CAR actually matters quite a bit for them to work properly as a tandem CAR. So for our preclinical study, we use Raji lymphoma cells. These are human lymphomas, naturally CD19, CD20 double positive, as is the case for most lymphoma cells. Wild-type Raji is double positive. We use CRISPR-Cas9 to engineer a CD19 negative mutant. And so for comparison, we have a mixed Raji group with 75% wild-type plus 25% CD19 negative tumors to mimic the situation where we already have mutant tumors growing out in the population. We give the tumors 5 days to engraft and then treat them with CAR T cells. Now, if the T cells were mock transduced, meaning they didn't express any CAR at all, it's an aggressive tumor model. Didn't matter if it was wild type or mixed Raji, the animals generally reach human endpoint within about 2 weeks. If we treated them with a conventional CD19 CAR, we appear to be able to control the wild type Raji, but not the mixed Raji. And that's exactly what we expected, since 25% of these tumors were CD19 negative to begin with. What was interesting to note was that if we waited long enough, the majority of the animals in the wild-type Raji group actually relapsed. And at the time of relapse, we can take the tumors out and analyze them for CD19 expression. And what we found was that even though they started out 100% wild-type corresponding to the red peak here, by the time they relapse, the tumor has significantly downregulated CD19 expression. This particular sample came from the liver of this particular mouse, but this was the general observation in these relapse animals, indicating that the Raji tumor model can actually capture spontaneous antigen downregulation, and that the conventional CD19 CAR T cells cannot protect the animals, just like they could not protect the human patients when the tumors lose CD19. Now, what we were very excited to see was that, if I can get this to move, animals treated with our bispecific CAR T cells can not only clear the tumor, but keep these animals tumor-free for a study that went on for nearly 200 days. And I will note, we often refer to this type of bispecific CAR as OR-gate CARs, because they can recognize a tumor cell as long as it presents either CD19 or CD20. You just need one or the other, which makes it harder for antigen escape to be successful, right? So based on this and other data, we decided to, and I have to say, with a lot of encouragement from senior colleagues like Owen here, and Tony Ribas and Don Kong, who really told me, like, this looks good. You should try to take it to the clinic. Because honestly, as an engineer, I was an assistant professor in chemical engineering at the time. How do you know that just because you cured some mice, that it's good enough to actually take it to the clinic, right? So really want to say thank you to all the senior colleagues who not only encouraged us, but actually shared all their protocols, allowed me to attend their group meetings to learn how to get this done. And certainly also providing actual resources, because these things are incredibly expensive to do. And so we decided to take this to the clinic. And this was the first investigator-initiated CAR T-cell therapy trial here at UCLA. As Rob mentioned, I'm a chemical engineer by training, not qualified to actually treat patients. Dr. Sarah Larson is our clinical PI and has been a fantastic partner in this endeavor. Here you see past and present lab members from my group who's really supported not only the IND preparation, but now the GMP manufacturing and the regulatory documentation. We also get a tremendous amount of support from the PIC Center on campus and the fantastic staff at the PIC Center. So to date, we have treated 11 patients on this trial. 10 out of 11 responded to therapy, and 8 out of 11, at 73%, achieved a complete remission. Among these patients, we have tested 2 dosing levels, 50 million CAR positive cells. That's a flat dose. We do not adjust dose based on patient weight. 5 out of 8 CRs and 2 out of 8 partial responses. At the second dosing cohort, we have 200 million CAR positive cells and saw 3 out of 3 CRs in our patients. Here you can see PET scans from a number of our patients. For those of us who are not accustomed to looking at PET scans basically image glucose metabolism, because tumor cells use up a lot of glucose, but so does the brain. So the brain will always light up, and PET scan is done with a tracer that's injected into the blood. So sometimes you will see accumulation of tracer signals in the heart and the kidney, and always in the bladder. So those are not tumor signals. All the other black signals you see basically indicates tumor presence. And so you can see our patients generally have very high tumor burdens. And this is not surprising, given that it's a first-in-human trial and there are already multiple commercially available products. And so the clinicians really have to decide why there should be a reason to put these patients on our trial, as opposed to recommending a commercial product, right? And so the fact that these patients have achieved such promising efficacy was very encouraging. What was even more encouraging, and quite frankly, a pleasant surprise, was how safe this therapy appears to be. And so neurotoxicity and cytokine release syndrome, or CRS, are the most common severe toxicities associated with CAR T-cell therapy. And to date, we have not seen any neurotoxicity of any grade in our patients. And the maximum CRS we have observed is grade 1. That's the lowest grade. So toxicities go from grade 1 to grade 5. Grade 5 is patient death. Grade 1 is the mildest grade. And so we're very happy to see that despite this very high tumor burden, which has been shown to be a predictor of severe toxicity, we have not seen this kind of response or toxicity. And I should say that, you know, up until this point, I think many in the field had come to accept that if you're going to get strong efficacy, you also just have to deal with the toxicity issue. But these data would suggest that there could be a way to engineer your T cells to achieve that efficacy without really pushing the patients all the way to the edge, right? So here you see the swimmer plot of our trial up until April of this year. So we at that point, we had a median follow-up of 20.5 months with median progression-free survival of 18.8. So this is quite a bit longer than what you had seen with the registration trial data showing 3 to 6 months of median progression-free survival. I will say that our patient numbers are still relatively small, and so do not want to sort of over conclude from this data, but we're certainly very happy for our patients so far. Now, one of our patients, and this is the only patient who achieved, among those who achieved a CR, that experienced a relapse. And this relapse occurred 18 months post-therapy. And so I'd like to share with you a bit more detail about this particular patient. So the patient was a middle-aged man with stage 4 follicular lymphoma with 4 prior lines of therapy. And all of this is actually quite typical among our patient demographics. What was unusual was that the patient had already previously participated in 2 other clinical trials. One that's Valosbio's anti-ROR1 targeted therapy, and another is Amgen's anti-CD19 bispecific T-cell engager. So BiTEs are basically bispecific antibodies where one arm binds CD3 to engage T cells and the other arm binds, in this case, CD19 to engage target cells, right? So the BiTE molecule can serve as a bridge that connects the tumor and the T cell. The patient unfortunately did not respond to either of those therapies, subsequently progressed on another set of chemo prior to enrolling in our trial. Now, at the time of screening, we could see B cells in this patient's peripheral blood, which is also quite unusual. And what we saw was that the B cells are CD20 positive, but CD19 dim, which is consistent with a lack of response to anti-CD19 BiTEs. What we were happy to see is, like most of our patients, within 60 days, which is the first response assessment point by our protocol, the patient had cleared his tumor. But as I mentioned, the patient relapsed at 18 months, and you can see from the PET scan tumor signal in the bone marrow and a few of the lymph nodes. By flow of peripheral blood, we can now see two different B cell populations, one that's 1920 double positive and one that is 20 single positive, CD19 negative. Based on this patient's clinical history and the fact that he actually did achieve a CR for 18 months, Dr. Larson decided that the best course of action would be to redose the patient with another set of CAR T cell therapy. And we fortunately had extra cells frozen down for this patient. And so with the FDA's approval, we redosed the patient and happily saw the patient return to a CR after 60 days. And the patient remains in CR at 12 months as of the last checkup in April. And so this is an example, I think, that really underscores the utility of having a bispecific CAR so that you can capture tumor cells that have already lost one of the antigens that the T cells have been engineered to target. And so we're very happy and quite fortunate to be able to bring this all the way to the clinic. But I am ultimately an engineer by training. And so what we're very interested in is how do we not only engineer one CAR that worked, but can we develop actually sort of generalizable design principles to engineer future CARs for other kinds of indications? And so as we engineered the 1920 CAR, we really came to understand certain aspects about, how the CAR interacts with the target antigen that we believe should be generalizable. And so to see if that truly is the case, my student Eugenia decided to take on another project where she engineered a BCMA CS1 bispecific CAR for multiple myeloma. And I will say that it took us about 3.5 years to build the 1920 CAR. And it took Eugenia by herself, with no sort of technician support, 15 months to go from DNA plasmid design all the way to finishing the first full-scale animal study that I'm going to show you today. Another student, Chris Nam, subsequently continued the project to round out the preclinical data. So I'll show you sort of the first full-scale study that Eugenia did. So we used, again, a heterogeneous tumor model, in this case, MM1S, multiple myeloma, naturally BCMA CS1 double positive. We used CRISPR-Cas9 to generate single positive versions of this line and gave a 1:1:1 ratio of this mixture. Day 5, we inject CAR T cells, and this is a very aggressive tumor model. So we end up redosing these animals with a second set of CAR T cells. And here you can see the distribution of our heterogeneous tumor model. In terms of controls, we included untreated animals, as well as 2 different bispecific CARs. Now, these 2 different bispecific CARs have the same BCMA targeting domain, but 2 different CS1 targeting domains. So we also tested each of the individual single input CARs. And what we can see is that the bispecific CAR number 2, shown here, clearly outperformed everybody else. And that then led to a significantly extended survival period. Now, when we submitted this for review, I think reviewers have come to expect that your animal models will just give you perfect in vivo data every time. So the question is, why did you not clear the tumor? Okay. And it turned out a lot of it has to do with cell manufacturing. And so I will also like to give a shout out to my fantastic manufacturing group, which supports our clinical trials. And it's a very hard thing to do. And so once you sort of optimize the cell manufacturing, you can actually quite reliably clear the tumor in the MM1S tumor model. And as part of our IND preparation, we compared our bispecific CAR against BB2121, which is now a commercially approved product known as Abecma. lymphoma. And in this case, we used a 100% BCMA positive MM1S tumor model, which means BB2121 should be able to recognize all the tumor cells. And you can see that our bispecific CAR still outperformed BB2121. And so we have submitted the IND for this just this morning. We had a call with the FDA. So we're hoping that we will get the IND cleared soon and be able to open this trial here at UCLA, again, in partnership with Dr. Sarah Larson. So now with the last part of the talk, I'd like to sort of switch gears and tell you about sort of more recent work we've been doing in our group. Now, CAR T-cell therapy has worked quite well for B-cell malignancies or HIM malignancies, but they've had a lot of trouble, quite honestly, with solid tumors. And there are a variety of reasons for that. One of these reasons is that solid tumors can create very immunosuppressive microenvironments such that immune cells, even if they manage to find the tumor, often become dysfunctional once they get to that microenvironment. And so tumor cells can do this through a variety of strategies. They can recruit inhibitory cell types, such as regulatory T cells. They can overexpress antigens for checkpoint receptors. And we became particularly interested in the soluble arm of tumor defense mechanism, in particular TGF-beta. So TGF-beta is known to be secreted or overproduced by a variety of solid tumors, as well as stromal cells in that tumor microenvironment. And in the context of a solid tumor, once it has already been formed, TGF-beta is a very potent immunosuppressive cytokine. So it can inhibit the function of not just T cells, but also natural killer cells and dendritic cells. And so we became interested in understanding, can we engineer T cells to overcome this particular tumor defense mechanism? And this was work started by a former MD PhD student, Yvonne Chen, with help from a former master student, Mickey Lorenzini. So what we wanted to do was two things. The first is we want the T cells to be able to inhibit endogenous TGF-beta signaling so that they don't become dysfunctional in the presence of TGF-beta, right? But we want to take it one step further and actually convert TGF-beta from a negative signal into a positive signal for our T cells. And the rationale is if solid tumors are going to overproduce TGF-beta, then a high local concentration of TGF-beta should serve as a signal that tells our T cells, you are likely to encounter tumor cells in this area and you should do something about it. Okay. Now, we're certainly not the first or the only people to have thought about rewiring T cell response to TGF-beta. There's actually a variety of strategies reported in literature. I would say the most clinically advanced of these is the dominant negative TGF-beta receptor, or DNR. This is basically a truncated form of TGF-beta receptor chain 2. It's missing the cytoplasmic signaling domain, so it can't signal. Now, the DNR can outcompete the endogenous receptor for TGF-beta binding, so it can serve as a sink. What it also does is because TGF-beta receptors form heterodimers, it's actually a dimer of dimers, it can poison that receptor complex. And that's actually why it's a dominant negative receptor. Now, the DNR is very good at inhibiting endogenous TGF-beta signaling, but it's a purely inhibitory signal. It cannot activate the T cell. And so it took us quite some time to try to figure out how do you do both of these things. And the answer may seem obvious in retrospect, which is to engineer a CAR that can respond to TGF-beta. And the reason this was not the first thing we tried was that no one up to this point, as far as we knew, had engineered CARs to respond to soluble antigens. Okay, so CARs are generally designed to target surface-bound antigens. And there had actually been prior papers reporting CARs that target surface antigens that can be shed. And those papers said the CAR did not respond to the soluble versions of those proteins. And so it wasn't obvious up front whether you could do this. But what we did know is that you can activate CARs with soluble monoclonal antibodies. Okay, so we figure it should be possible. And so we had to figure out how. So as sort of a proof of concept or sanity check, we took the very well-behaved CD19 CAR T cells and tried to activate them with soluble CD19. And what we found was that they do, in fact, express activation markers in a dose-dependent manner in response to soluble CD19. This is just the ectodomain of CD19 secreted by HEK293 T cells. Furthermore, these T cells actually secrete cytokine in response to soluble CD19, just as they do in response to surface-bound CD19. And this is important because there's a difference between proximal signaling and downstream signaling in terms of T cell receptors and presumably CARs. And we want to make sure that we actually get the downstream effector functions in response to soluble ligands. Now, we did quality control on the CD19 proteins, and to our surprise, instead of a monomeric band, which is what we were expecting, on a non-reducing gel, the dominant band was actually a dimer, okay? And this is most likely an artifact of how we make these proteins. Perhaps they're too concentrated. In fact, you can see a whole ladder of multimers running up the gel. But the fact that the dominant species is a dimer, together with what I mentioned before, which is monoclonal antibodies, which are bivalent in nature, can activate CARs, made us wonder whether we just need the ligand to be a dimer for this to work, right? So then we did this test where Zenon built a GFP binding CAR and tried to stimulate these CAR T cells with either monomeric GFP or dimeric GFP. I will note the dimeric GFP was a generous gift from Todd Yates' group in the chemistry department here. And what we found was that only the dimeric form of GFP could activate our T cells. So then the question becomes why? Why do you need the ligand to be a dimer? So when your ligand is a dimer, what in principle can happen is subunit number 1 can bind to T cell number 1 and subunit number 2 can bind to T cell number 2. And so the ligand can essentially serve as an adapter, just like the bites that I mentioned to you earlier, right? So you're pulling 2 T cells together. Now, if this is the only reason why the dimeric ligands work, then we could be in a bit of trouble because you need a high local concentration of CAR T cells in order for this dimer sort of pulling together to occur, right? And that may not be so easy to do in vivo. So we wanted to see whether you really need this cell-cell contact. And so to test that, we did an experiment where we loaded the T cells with FLO4, which is a calcium indicator that cells would then light up whenever there's a calcium influx, which is one of the first things that happen when T cells get activated. And then we add GFP at time 0, and we can find some of these cells lighting up without ever coming into contact with another cell, okay? So this tells us cell-cell contact is not essential. So then why do you need it to be a dimer? Another possibility is that when the ligand is a dimer, you can pull together 2 receptors, not from 2 different cells, but on the same cell surface, okay? So maybe what you need to do is trigger receptor dimerization. So to distinguish whether it's really the ligand dimerization that's important or the receptor dimerization that's important, Zenon built a second set of GFP CARs, and these CARs bind a different epitope on GFP. So what this means is if we co-express the 2 different GFP CARs on the same T cell, then in principle, we can trigger receptor dimerization even when the ligand is a monomer, because the 2 receptors can bind to 2 different spots on the same GFP. And indeed, what we found was that if we express either CAR alone on the T cell does not respond to monomeric GFP. But if you co-express the 2 CARs, then it responds to monomeric GFP. And once again, this response does not require cell-cell contact, okay? And so based on this and additional data, we decided that it should be possible to build CARs that respond to soluble antigens, provided that ligand binding can trigger receptor dimerization. Okay, so then we come back to the question of how do we build a TGF-beta targeting CAR? And luckily for us, TGF-beta is naturally a homodimer. So we only needed to build 1 CAR instead of 2 for this dimerization to occur. And so I'm going to show you some data using a CAR that contains a CD28 co-stimulatory domain, but it also works with 4-1BB. And in fact, later on, I'll show you some data with 4-1BB CARs. And so the first thing we wanted to make sure of is that the TGF-beta CAR can actually do at least what the dominant negative receptor does, which is to inhibit endogenous TGF-beta signaling. Now, when TGF-beta signaling occurs, it triggers a cascade that involves the phosphorylation of SMAD2. And so what we can see is that the more TGF-beta we feed, normally you would get a darker phospho-SMAD2 band. But if the T cell expresses a functional TGF-beta CAR, we can basically abolish that signaling cascade. This is purely by out competition. We did not knock out the endogenous receptor. Now here we show 2 different negative controls. EGFRT is just truncated epidermal growth factor receptor. It serves as a transduction marker. scFv-less CAR is identical to the TGF-beta CAR, except it's missing the ligand binding scFv domain. And the reason we like this as a control is that way we know for sure the difference is due to ligand binding as opposed to overexpression of signaling domains like CD3 zeta or CD28. We see the same behavior in both CD8 and CD4 T cells, indicating the CAR is quite good at essentially just outcompeting the endogenous receptor. So then we want to know whether we can do step number 2, which is to activate the T cell in response to TGF-beta. And here you can see once again the observation of upregulation of activation markers, specifically in TGF-beta CAR T cells in response to TGF-beta. And we can again observe production of Th1 cytokines in response to TGF-beta. And very importantly, this response is dose dependent. The more TGF-beta we fed, the higher the cytokine production levels. And this is important because we don't want these T cells to just be activated anywhere in the body. We need them to be activated specifically in areas of high local TGF-beta concentration. I will note, because the receptor is a dimer, there is actually a hook effect. So if you go very high up, the curve will start dropping. But the drop doesn't actually occur until you're at way over physiological levels. So we don't think that would be a concern. Now, on the same note of safety, I will say that the CAR only responds to the active form of TGF-beta. It does not respond to the latent form. And the latent form is what you would generally find in healthy tissue. So latent TGF-beta gets converted into mature TGF-beta through metalloprotease processing, and metalloproteases are found at much higher concentrations in the tumor microenvironment, right? Now, one of the things that TGF-beta normally does as an immunosuppressive cytokine is it inhibits T cell growth. And so we can see that with our negative control SC avilis CAR T cells, if we culture them under standard ex vivo conditions with cytokine support, but also add TGF-beta, we can strongly inhibit T cell expansion, okay? In contrast, TGF-beta CAR T cells grow in response to TGF-beta. I will note the y-axes are different scales on these plots, okay? So TGF-beta CAR T cells respond to TGF-beta as if it's a growth-promoting cytokine. We did this in multiple donor cells just to make sure it wasn't a fluke. And once again, this promotion of T cell expansion is dose-dependent. So the more we fed, the more TGF-beta we fed, the better the T cells grew. In fact, the T cells were growing so well that my student Zenon got a little bit worried that maybe we had somehow transformed these cells. And so he did an experiment where he cultured the cells with TGF-beta for 3 weeks, and then he stopped feeding TGF-beta and cytokines. And once he did that, the culture would crash. Okay, and so this is just to make sure that we didn't somehow immortalize these cells through this process. And so these data showed us that TGF-beta CARs can effectively convert TGF-beta from a suppressive signal into an activating signal, resulting in cytokine production and T cell proliferation. But one thing soluble TGF-beta does not do is it does not trigger cell killing. Okay, and we think the reason is when the ligand is a soluble antigen, there is no immunological synapse. And therefore, there is no actin reorganization that allows for directional release of the cytotoxic granules. And this is actually quite important, again, from a safety perspective, because you don't want the T cells to just be spewing out cytotoxic molecules whenever there's TGF-beta around. You want it to be directed at tumor cells. But then how do you do that? And if you don't do that, it's actually not all that useful as a cancer therapy, right? And so the way we do this is by going back to our work on bispecific CARs and build these tandem CARs that can simultaneously target a surface-bound tumor antigen and convert TGF-beta into an activating signal. And so the concept here is now we would have these T cells that can go into the tumor microenvironment, specifically recognize the tumor, but also start modifying that environment by essentially serving as a sink or soaking up TGF-beta and converting that into the production of immunostimulatory cytokines, which can then hopefully further stimulate endogenous immune cells in that environment. And this was really work then performed by Andrew Howe, another PhD student who graduated this past year. And so we decided as an indication to focus on glioblastoma because it's a clear unmet need. It's an incurable disease. And I think it's gotten actually quite a bit of press because of prominent figures in our nation, people like Joe Biden's son, John McCain, Teddy Kennedy, who all died from this disease. And we decided to pair TGF-beta together with an antigen called IL-13 receptor alpha 2, which is present on some glioma patients' tumors. And so in building a bispecific CAR, you need to first make sure it actually is bispecific. And so we made sure that in response to TGF-beta, we see, again, activation from these bispecific CARs in the middle cluster here. This is in contrast to the single input IL-13 receptor alpha-2 CAR, which does not respond to TGF-beta. And once again, the DNR, that's the dominant negative TGF-beta receptor, does not activate the T cell, right? So if you co-express it with a single input CAR, you get the same output as the single input CAR in response to TGF-beta. So does it respond to IL-13 receptor alpha-2? So in this case, we co-incubated our CAR T cells with patient-derived glioma cells. And then here we stained our T cells with cell trace violet, or CTV. Now that's a dye that dilutes every time the cell divides. So the more times the cells divided, the lower the CTV intensity. And what we can see is after a 94-hour co-incubation, the bispecific CAR shown in yellow here can kill just as well as the single input CAR with and without the DNR. However, these bispecific CAR T cells divide more. So we see a lower CTV intensity, as well as a higher CAR T cell count at the end of this co-incubation period. Okay, so does this translate to actual survival advantage? So in CAR T cell therapy, the standard models are human xenograft models done in NSG mice. So here we took, again, human patient-derived neural spheres, injected directly into the brains of these mice, and then 7 days later, treated with, again, an intracranial injection of CAR T cells. I will note intracranial injections of CAR T cells have been done in human patients. It is safe, and there are lines of evidence suggesting it may be more efficacious than peripheral infusion in the context of GBM. And so here we can see the bispecific CAR shown in orange outperforming the single input CAR shown in purple. And this single input CAR is actually co-expressed with the dominant negative receptor. At the doses we used, a single injection of 500,000 CAR positive cells, the single input CAR plus DNR basically didn't do anything, whereas the bispecific CAR can significantly extend survival. Now, as I mentioned before, a major sort of goal with the TGF-beta CAR was to modify that tumor microenvironment. But that's a difficult question to ask in an immunodeficient mouse like NSG. And so we wanted to test this in the syngeneic mouse model. And very luckily, human and murine TGF-beta are actually highly conserved in terms of amino acid sequences. And so here we took human TGF-beta CAR T cells and exposed them to either human in blue or mouse in orange TGF-beta. And we see that the response curves in terms of cytokine production is basically identical. Okay, and so the TGF-beta CAR as is can be tested in a syngeneic murine T cell model. And so what we did here was take CT2A murine glioma cells. We actually introduced human IL-13 receptor alpha-2 into these CT2A cells. And then 6 days later, inject the animals with CAR T cells. And 7 days after that, took the tumor-bearing T cell-bearing brains out, generate single cell suspensions, and perform CyTOF analysis. And this is done in Zoran's core. So also a shout out for a lot of help in Begonia here, who really helped us putting this panel together. Now, in this experiment, let me walk you through the legend here. S-CAR is the single input IL-13 receptor alpha-2 CAR. S-CAR plus DNR is the single input CAR plus the dominant negative receptor. 10-CAR is the tandem CAR for IL-13 receptor alpha-2 TGF-beta. It was very wordy, so that's our acronyms. So treatment with any of these CAR T cell groups resulted in a substantial decrease in monocyte-derived suppressor cells. Furthermore, we see an increase in T cell content, and notably, the bispecific CAR treated group had the lowest MDSC and the highest T cell content. So if you actually plotted the ratio of T cell to suppressor cells, it's substantially higher for the bispecific CAR T cell treated group. We wanted to then take a deeper look and perform single cell RNA sequencing. The data here were analyzed by our current MD PhD student, Ryan Shi, in the lab. And what we can see is that, again, treatment with any of the CAR T cell therapies resulted in a substantial decrease in tumor content, as you would expect. What we also see is that treatment with bispecific CAR T cells in particular resulted in a significant decrease in peripheral macrophage population. That means instead of sort of brain resident microglial cells, these are macrophages that likely came from peripheral circulation. There, I will say, are sort of conflicting literature on precisely what these peripheral macrophages do, but generally they're considered to be not a great thing for immunotherapy response. And so we think this reduction is a beneficial effect. Now, if we focus specifically on these macrophages and look at their phenotypes, what we also see is that the macrophages present in animals treated with our bispecific CAR T cells have higher levels of CD83, which is sort of considered an activation marker indicating a more immunostimulatory phenotype as opposed to immunosuppressive phenotype of macrophages. The group treated with bispecific CAR T cells also had the lowest level of TGF-beta induced proteins, as well as various galactans that have been shown to be counterproductive for immunotherapy response. And so these data suggest that treatment with our bispecific CAR T cells can actually modify not just the T cell compartment, but also the myeloid compartment in the brain into what is potentially a more favorable environment. But of course, this is a T cell therapy, so we're also interested in what's happening with the T cells. So what Ryan did here was he regressed out all the non-T cell events and reclustered the remaining T cell data points. And so now he can see a much sort of more detailed view of the different subtypes. And what we saw was that among the CD4s, animals treated with our bispecific CAR T cells have a much larger proportion of activated T cells as opposed to regulatory T cells, which are suppressive. Among CD8s, we see a large population of this innate-like cytotoxic T cell population that was recently described by Ming Li's group at Memorial Sloan Kettering to be associated with favorable responses to therapy, right? So once again, we seem to be seeing signals that indicate treatment with our bispecific CAR T cells can alter the immune cell composition. And I will say that almost all of these events did not show CAR expression, at least not that we can detect. And so what we seem to be seeing is a change in the endogenous T cell population in the brain. So did this translate to actual benefit in survival? So again, this is our CT2A tumor model. The bispecific CAR shown in pink here show better tumor control compared to the single input IL-13 receptor alpha-2 CAR, resulting in a significant increase in survival. I'll note these p-values are always adjusted for multiple comparisons. It's a sore point because the reviewers are picking on p-values of 0.013. And so the question is, well, I think the data quite clearly show, right, in both xenograft and syngeneic mouse models that the bispecific CAR outperforms the single input CAR. And so in collaboration with Rob and Dr. Linda Liao, we're seriously planning out how to take this to the clinic for patients here at UCLA with GBM. But, you know, as a matter of scientific interest, we also wanted to know how did the bispecific CAR do in comparison to the DNR group, right? So as a reminder, we had previously tested the single input CAR plus the DNR in the xenograft setting. And in that setting, the DNR didn't do very well at all, okay? Interestingly, in the syngeneic mouse model, we see this sort of bifurcated response. So if you pay attention to our negative control, SCFE-less CAR T cells, the animals generally get what we would describe as uncontrolled tumor progression. The tumor curve just goes up and then the animal dies pretty quickly. And so if you look across the treatment groups, it's very clear that the bispecific CAR had fewer animals that had no control of the tumor whatsoever, whereas the DNR group actually had about half of the animals falling into that group. What's also interesting, though, was about a quarter of the animals in the DNR group actually ended up with tumor-free long-term survival, okay? And so this is what I mean by a bifurcation or bimodal response from this group. And because of that quarter tumor-free survival, there actually was no overall statistically significant difference in the survival curves, okay? And so I think there could be really interesting immunology questions going on here. As I mentioned before, here we introduce human IL-13 receptor alpha-2 because we wanted it to be representative of what happens in the clinic. But another student in the lab, Justin, recently demonstrated that the CAR actually also recognizes mirroring IL-13 receptor alpha-2. So we're now performing studies for various projects in the lab with the mirroring system, and we may see whether that ends up changing these patterns or not. So with that, I would like to thank again the organizers for having me present here, and also the people who actually do the work. Quite frankly, now my lab members don't even trust me with a pipette anymore. So all of this work is really done by the hardworking people in the lab. And also it takes a lot of money, quite frankly, to do the kind of work we do. And so thank you to all the funding agencies, and thank you for being here. And I'm happy to answer any questions.
Could TGF beta biphasic CAR generated outside the body to make it quicker in vitro and then injected back as in the tumor vicinity to make it quicker?
Ah, okay, yes. So let me repeat. Do I need to repeat the question? Can they hear on Zoom? No. I can just, okay. So we do generate these CAR T cells outside the body. There are actually a lot of people trying to figure out how to do it inside the body, but right now we take the T cells out and we make them outside and then put them back in. I think the second part of your question is, can you just put it near the tumor as opposed to in the blood? So in the context of glioblastoma in brain tumor, we are in fact putting it directly into the brain. Now, in the brain, there are a couple options. So the neurosurgeon will always try to cut out, surgically remove as much tumor as they can. So that leaves behind a hole. It leaves behind a cavity. So we can inject the T cells directly into that cavity. Alternatively, we can inject the T cells into the ventricle so that it gets to circulate with the cerebral spinal fluid, and it may be able to reach sort of metastatic sites more efficiently in that way. But this is something that we and many other groups are actively looking into in terms of what would be the best injection route to achieve efficacy.
You mean you haven't done it yet?
Not in GBM. So in lymphoma, we have done it in human patients. But in GBM, we're just planning the trial right now.
So this is a good option for a quicker control of the tumor. Is that right?
Well, we're hoping that it would be a good option for good control of the tumors.
For the going back to the CD19, CD20 dual CAR, the patient who relapsed, in all those patients, were you seeing at 30 days that your CAR was sort of depleted, and that's why there was no actual dimeric CAR left over at that 18 months?
Actually, very interesting question. So we've only had one relapse case so far. So this is an N of one. But what happened with this patient was we tracked the T cell presence by droplet digital PCR, which is, by the way, done by Bea in Don Kong's group. So also a shout out. We can, for this particular patient, we could see T cells up to 9 months. And then past 9 months, at 12 months, the signal became undetectable. However, at 18 months, when the patient relapsed, the signal came back. Okay, so it suggests that the T cells were still around. They were just not around at high enough numbers, at least 9 peripheral blood, for us to detect between 9 and until 18 months. But when the tumor came back, they seem to have then expanded so that the signal came back above detection. But clearly, it was not enough to actually control the tumor. So then when we reinfused, we saw again sort of an increase, but the second peak of expansion was not nearly as high as the first peak. And it could be because the second time around, the tumor burden was much lower compared to the first time. It could also be that the body had developed some level of resistance so that the T cells could not expand as much. We don't know which of the two, or maybe both, are in play. But in this particular case, it appears the T cells, the first injection of T cells, were still around at detectable numbers. They just couldn't clear the tumor.
And sort of the same question for the glioblastoma one at the end. Did you see any difference between the persistence of the CARs between the DNR or the dual?
Yeah, great question. So in the GBM model, it's a little bit tricky because it's intracranial. So we can't do time point, like we do RO bleeds, but we basically don't see any significant number of T cells in the periphery. And so the only way to detect T cells is sac the mouse and then analyze the brain. And when we do that by RNA-seq, we actually don't really see like any significant number of CAR positive T cells based on RNA-seq. Now, we're not completely sure yet whether it's a flaw in our analysis or if the T cells are truly absent. But at this time, I don't have data to be able to say whether the T cells are, the CAR T cells are present at higher numbers in the bispecific case. We do know the total number of, or at least the fraction of T cells is higher in the bispecific CAR T cell treated groups, but they may not be CAR positive T cells.
But that's at euthanasia time point.
Yes.
Cool talk as
Thank you.
usual. Quick question about the TGF-beta specific CAR T. Does it have any effect on wound healing? Because areas of wound healing would have a lot of TGF-beta.
Yeah, great question. So mature TGF-beta is found at sort of elevated levels at wound sites in the gut and tumor microenvironments. So we have not specifically tested wound sites, but we have done sort of full body necropsies of mice, and the pathologists have not noted any sort of gut toxicities. So we're hopeful that perhaps it's because it's dose responsive, right? Like you actually need quite a bit of active TGF-beta for the CAR to be activated. Perhaps that's why we're not seeing toxicity. But quite honestly, when we take this to the clinic, safety is because you can see perfectly safe signals in the mouse and then something happens in a human. And so suicide genes are definitely something that we would consider. In the context of GBM, in consultation with Linda and others, the sort of message we're getting is that specifically in the context of GBM, high dose steroids is sort of routinely used to control inflammation. And so if toxicity were to occur, that likely would be the first strategy rather than trying to use a suicide gene. But if we were to go for any other indication, I think a suicide gene likely would be a good idea, just to make sure you have a way to counter any adverse events that are unexpected.
Cool. Thank you.
Thanks.
Hi, thanks. Is this, do I just talk?
Oh,
into the
yes.
box like this? Yeah. Okay. Thanks for a great talk. I have a question on like a future looking question for the clinical trials in GBM. I know they have DC pulsed vaccines for GBM, and I'm wondering, yeah, from Rob. So I'm wondering how you envision that technology and or the CAR Ts for GBM working in the clinic in the future. Like a partnership, or one being first line, one being second line? What are your thoughts?
Yeah, so I can give you my thoughts and then maybe Rob can chime in if he has additional comments, because I really am not an expert in the DC vaccines. So as a first in human trial, we likely would keep it separate just because you don't want to overcomplicate what you're testing, right? You want to be able to clearly say the safety signal belongs to this therapy, and if there's any efficacy, how to attribute it. I think combination therapy, be it DC vaccines or checkpoint blockade or other types of immunotherapy, are definitely things we should consider in GBM, because GBM is a very heterogeneous tumor. And so targeting a single antigen likely will not get the job done. We're hoping that with the TGF-beta strategy, we're not just targeting IL-13 receptor alpha-2, but also modifying the immune microenvironment so that we can induce an endogenous immune response. It remains to be seen whether that actually is accomplished, but that's the hope, right? But I think combination therapies can certainly be another way to provide that reinforcement, if you will. Rob, do you have?
I mean, I think it comes down to what the vaccine is going to add to it. Like, we'll do a vaccine together with systemically administered T cells, and then it will expand the T cells in vivo. For this, where you have a local infusion, they never make it out. It's hard to know how that's going to be, and what it's going to add to the system unless you add them into the tumor, and that's not as well established in the sense. But sometimes giving a vaccine that promotes a more diverse, where you can get more epitope spreading, and maybe you can think that. Although I think the NIH reviewers didn't seem to like that component.
The precise phrasing is it's a kitchen sink approach. Yeah.
So that's not part of it any longer. One more.
Hi, how are you? This is probably a naive question, but instead of like a bispecific CAR T, have people tried like libraries of different CAR T targets?
a library of different.
Right, like of monospecific.
So instead of a tandem CAR, can you
Yeah.
just put in, okay, yes, so you can do that. In fact, I even have slides that can show you that. So there you go. Okay, so if we're just talking about bispecific, of course, you can extrapolate this to multi-specific, right? We built tandem CARs. You can put two different CARs into the same cell. We call that dual CARs. You can have two different cell products and then pull them together. Okay, and the reason we so CAR pooling is generally speaking the least favorable, because now you have to make two products. That's super expensive. You sort of double the probability of failing to generate the product for a given patient, so on and so forth, and they have to compete against each other. The dual CAR situation is challenging because now you have to fit in a lot more DNA per cell. So you have to repeat at least the extracellular spacer, transmembrane, and cytoplasmic signaling domains, right? And so now you have to fit a lot more DNA bases into your viral vector, which reduces the transduction efficiency, which then reduces the potency of the product. And we've actually done head-to-head comparisons in the context of BCMA CS1 CARs, comparing tandem CAR versus dual CAR, and the tandem CAR is clearly superior to the dual CAR. But I will note that not every antigen pairing is suitable for a tandem CAR architecture, because depending on the target epitope, where it's located relative to the surface of the target cell, you may end up with steric clashes that you cannot overcome with a tandem CAR. And in that case, a dual CAR may in fact be superior just because you can't get the tandem CAR to the right shape, right? Yeah. Mm-hmm.
One last one.
Actually, the tandem CAR was exactly the same question that I was trying to ask.
Okay.
Maybe one last question, I think is more philosophical one.
There is a Q&A on
Do we?
online.
Okay.
So Patrick Chang asked, basically, did you ever figure out why there was less toxicity? And were any patients previously treated with CAR T cells? So good questions. Answering the second question first, our protocol originally excluded all patients that had previously received CD19 CAR T cell therapy. We did not exclude CD19 targeted therapies in general, but excluded CAR T cell therapies. And the reason was we wanted to make sure that what we see is attributable to our product and not some other product. But as I mentioned, we had a patient who relapsed and was redosed, right? And as part of that amendment, we actually amended the protocol. So now we can admit patients who have previously received CD19 CAR T cell therapy, although so far we have not yet treated one. In terms of why there's lower toxicity, I can tell you my hypothesis. I have no proof in terms of whether this is indeed the reason. So we engineered the CAR to be efficacious. We did not engineer it to be safe. And that's in part because there's no good mouse models for safety assessment of CAR T cells. And so I think the safety really is most likely a byproduct of how efficacious the cells are. So most of our patients, as you may recall, only got 50 million CAR positive cells, and that's a much lower dose than any of the commercial products. And yet these sort of small numbers of starting CAR T cell population appear to be sufficient to get rid of the tumor cells, right? And what we have also noticed was that the T cells in our patients can expand dramatically. So eventually they reach very large numbers, but they always peak at day 14 post-infusion. Okay. Now, in contrast, the tumors seem to disappear much faster than that. We don't have good ways of quantifying tumor sort of presence in early time points, because PET scans done too early can give you false signals, and you often just don't see B cells in the peripheral blood, even when the patient clearly has tumor, right? But what we could see was some of our patients had so much tumor that you can feel them. They had palpable nodules in the neck, in the abdomen. And whenever there were palpable tumors, they would disappear within 14 days. And so that suggests to us that the tumors were coming down very rapidly while the T cells were going up. And because the T cells expansion is a bit delayed compared to most commercial products, we think we end up with a temporal separation where by the time you have a ton of T cells around, there's not a whole lot of tumor cells left to sort of dramatically activate that huge T cell population. And perhaps that also contributed to the lower toxicity. And the final thing I will say is when we manufacture these cells, we enrich for naive and memory T cells. And these cells generally have a softer touch, if you will, right? They're known to be a bit more persistent, a bit less intense in terms of their immediate effector response. And perhaps that also contributed to the lower toxicity. But I will say all of this is speculation because we don't have a head-to-head comparison of, naive memory T cells versus cells made from PBMCs, so on and so forth. But these are sort of the lines of evidence that we're seeing that suggests what might be happening here.
All right. I think we'll let it go with the last question. Thank you very much.